Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Elisa Canepa, an assistant scientist at Temple University. Elisa is also a 2022 Toffler Scholar. We will be discussing her experiences as a neurobiologist investigating cerebral vascular dysfunction. So let's dive in. Welcome.
Hello, Deborah. Thank you for having me. So happy to be here.
I'm excited for you to be here. You're in a really fascinating area of research, this kind of convergence between the cardiovascular and the brain. And there just seems to be more emphasis in this area. And so I'm very looking forward to hearing what you're doing and some of the breakthroughs that you might be seeing in the next few years. So I'd like to just start with what inspired you to pursue a career in neuroscience?
Well, Deb, brain, we're talking about the most fascinating organ we have. Well, that's if you think that it makes what you are, and so your thoughts, your perceptions, your memory, that's what your brain does. And unfortunately, sadly, in Alzheimer's disease, all these things are lost because of Alzheimer's disease is a bad detrimental neurodegenerative disease affecting memory loss, and not only. Thus, this was to me fascinating. How does it works and the molecular mechanisms that can trigger such bad toxic event and ultimately leading to the neuronal demise. That to me, it's fascinating. Yeah.
When were you aware of this fascination? Was it when you were younger? Did this happen sometime in, through college or graduate? When did it become, oh, okay, I want to dedicate my life to this?
Through college, I became passionate to brain, to neuroscience. I started approaching neuroscience as a behavioral science, and then with my PhD studies, I dig in the molecular mechanism inside what's going on. And then during college and PhD, I thought, I understood that was my path for sure.
So you're in Dr. Silvia Facetti's lab, I believe, and you're the lab is really focused, like I said, on this understanding cardiovascular and the brain. But your research is really focused on this thing called CAIs. What is a CAI, and what is the role in Alzheimer's treatment?
Okay, CAI are carbonic anhydrase inhibitors, are drugs. It's a big term for comprehending several drugs that all inhibits carbonic anhydrases, which are enzymes inside our cells. Specifically, carbonic anhydrases, like the one that we have been studied, metazolamide and acetazolamide, have been used for decades. At the beginning, they were used as diuretics. Then later on, they started using them for high altitude sickness, edema related to high altitude, or for glaucoma. Therefore, understanding that behind it was a vascular function. And then our lab discovered that this specific acetazolamide and metazolomide carbonic anhydrase inhibitors block specific cell death pathways in neurovascular cells, therefore including in neurons, including endothelial cells, which are the cells that form the blood vessels, including neural cells, which are the ones that are helping the blood vessel to contract, like smooth muscle cells and pericytes, or glial cells like astrocytes and microglia, which are the cells that normally regulate the inflammatory states of the brain, and also having this dual function, communicating and regulating the vascular tone, neurovascular coupling. That's how it's called. And we lately discovered that carbonic anhydrase inhibitors never used in Alzheimer's disease related conditions are indeed beneficial in Alzheimer's model. And that's where what I found, and I published very recently in the Alzheimer and Dementia Journal, that not only the carbonic anhydrase inhibitors rescue cognitive decline, but also ameliorate neurovascular dysfunction and neuroinflammatory states. And that's a very big thing, and let's see how it's gonna end up with carbonic anhydrase inhibitors employed specifically in NAD field.
So you said rescue the cognitive function. Is it reversing the disease, or is it slowing down the disease? What is that?
Yes. What we do is performing several behavioral tasks with animals. That's what I'm working with. And we employ tests as barn maze that assess the spatial memory. And for example, obviously the animals do not learn from one day to the other. You have to train them. So basically after a couple, 5 days of trainings where you teach them where to reach, for example, a so-called escape hole. So you are teaching them where to go to not be bothered by the external environment. So you trigger them to find this escape hole where they know that they are going to find peaceful environment. So that is the goal, is that to let them learn to find the escape hole. And what we measure, for example, in the probe day, which is the final day after the training days, we measure the mistakes they make for finding the escape hole. We measure the distance they make. So the more distance they travel, the more they are trying to figure out where the escape hole is. Therefore, the more cognitive impaired they are, the longer distance they're going to have compared to the wild type, the more mistakes they're going to make compared to the wild type to find the escape hole. On the opposite side, we have animals that undergone carbonic anhydrase treatment that have completely, or let's say completely, very highly reverted spatial memory. And that from there, we wanted to understand which were the biochemical features inside the brains that were triggering that behavioral amelioration. And that's where it started the project on the Swedish Dutch Iowa animals. That's how is the animals model called.
So that's really interesting. I'm wondering, and I don't know if there's any connection, but there's people that live in high altitudes and they don't need high altitude sickness, or medicines. They have, different kind of hearts, different kind of, their hearts are stronger if you're living at, 18,000 feet or 16,000 feet. Is there any connection there that we're learning with the group of people that actually don't need those kind of drugs, but maybe there's learning from how they develop organically or environmentally to this disease?
Well, for sure, the people that live at such a high altitudes have a different blood pressure compared to people that live at the sea level. Also dimension of red blood cells are different. They are much bigger in people that live at high altitude because it's easier for them to get for carrying more oxygen. So for sure, and therefore the dimension of the blood vessels is different. For sure we can understand those beneficial mechanisms that occur in a normal high altitude environment for then applying those beneficial mechanisms in the pathology of Alzheimer's disease, right? So, yeah.
So the drugs that you're studying and its relationship to Alzheimer's is understanding kind of those mechanisms, if I understand that correctly, the oxygen flow, the way the blood vessels may contract or expand, the way its relationship to inflammation in the brain. Am I understanding that correctly?
You're understanding correctly, Deb. But as you know, science is not just Dr. Canepa is doing all these things that you just mentioned. I am observing and let's say I'm more focused on endothelial cells activation, therefore blood-brain barrier dysfunction, loss of vascular integrity in the brain that is linked and totally associated with the neuroinflammation. And there are people like one of my collaborators in Denmark, Dr. Gutierrez, that, for example, is an engineer, and what he performs is more functional studies, more, let's say, optical physics studies on cerebral blood flow, cerebral blood volume. So there is that part of the study that include the functional study and the oxygen levels, etc. We have, for example, I'd like also to mention some other collaborator we have at University of Florida, Dr. Prokov, which is the director of the Neuroscience Tissues Bank of University of Florida, and is helping us with very cutting-edge techniques, which is called RNA-scope. So he does the part of the transcriptome, meaning all that part that it's no more proteins, the proteomic part, which is the one that I mainly study. It does the genomic parts, specifically the RNA scope. It's a very beautiful technique because it's not just understanding how much RNA is expressed. That's something that another very cutting edge technique that does this is the RNA scope for the RNA-seq, for example, RNA sequencing. This RNA scope instead, besides telling you which RNA is present, it tells you in a brain slice where is it expressed. So, for example, the carbonic anhydrase, which are the enzyme that we study in the blood vessels, and if this expression is less or not influenced by the presence of amyloid beta or tau, which are proteins that we know that accumulate in the brain of Alzheimer's disease patients.
So this whole area of understanding this blood-brain barrier and kind of that interface is fairly new. I don't think we were studying this, a long time ago. It's getting more emphasis in the last decade. So that's kind of a significant finding in itself. What other kind of significant findings are you seeing just kind of maybe emerging? I mean, you're seeing some of these drugs and how they're maybe even reversing some of the destruction, if you would, inside the brain. Are there other kind of breakthroughs that you're very excited for as you're doing your research?
Absolutely, yes. And I think that one big thing nowadays, besides those two techniques that I was talking you about, RNA sequencing and RNA scope, the big thing, in my opinion, that we're gonna see in the next decade is gonna be biomarkers, meaning the prediction is the big thing what we are going. Obviously, if we see the speed that we are going on with artificial intelligence, for example, or we can imagine artificial intelligence combined with machine learnings and with the elaboration with this big data set of biomarkers, I was saying, but let's say also imaging results or genetic information. We are going to go in that direction. And obviously, in the future, there are going to be much more easier, much more sensitive assays. And I mentioned biomarkers, but biomarkers are going to be obviously mandatory in parallel with imaging. Therefore, we know that in the future, MRI studies, PET studies are going to be much more sensitive, and this is going to be much more helpful to predict, although the lack of neuropathological events that can be obviously amyloid of tau accumulation, but we were saying neurovascular dysfunction, neuroinflammation. We know that there is mitochondrial stress, oxidative stress. Let's say that these are already features that are expressed. We want to understand what is coming before for then preventing what is going to come in next. And yeah, that's my opinion, biomarkers and in combination with imaging approaches.
Yeah, there's a lot going on in the imaging too.
Vessel person, for me, like imaging functional, besides our fundamental, obviously, study of cell molecular biology, is then understanding, are they functioning? Is the cognitive impairment ameliorating? We need to also understand the outcomes. And then it's also the big debate with the current FDA-approved drugs. But we can talk it later.
So what are some of the biggest challenges in doing this kind of advanced research? Because it is new. It is kind of risky sometimes, for people to be giving you funding or to give you. What are some of the biggest challenges that you kind of have in doing this kind of research?
Okay, there are the experimental challenges. It's part of the basic science research, and let's say that's a combo with the frustration, no? But as far as there is curiosity, that frustration is never sufficient to put you down yet, let's say. And I would say the challenges are you need to connect. You need to be able to, once you attend conferences, you need to try to recharge the people that can give you a different point of view from your research, people that have different skills than yours, I was talking about my collaborators. So we're approaching the same neurovascular dysfunction from different point of views, and to me that's also another big thing in the future, the collaborations, and that's what you guys are helping us doing as well, because it's not the single molecular biologist that is going to make the difference, but it's going to be him with a clinician and the bioinformatician, like a team of scientists that can give his competent point of view.
Yeah, the collaborations are really important. And you've talked about some of the interdisciplinary. You're working with an engineer in imaging. You're working with somebody that is working on the RNA sequencing and scoping. That's a very different technology and technique. You've also mentioned, in the future, you're going to have to be working with people that are for more advanced imaging or even computational. And, that really puts a stress on a researcher to make those connections and keep them there and work those. Yeah, I can definitely see how that's difficult. You're kind of early in your career. You're advancing in your career, and there's several kind of promotions in your future that you're hoping for. One of the transitions that you'll make is to someday have your own lab. Have you given that any thought as to kind of what you want to focus on in the future? Are you still kind of in that area of, I need to do this science, I need to learn from my mentors? I'm just kind of curious as to, are there advancements that you over your career want to kind of focus on?
Deb, I want to be 100% sincere with you. To me, what is important is, we were talking about this before, the awareness of Alzheimer's disease, like the communication about what's going on with a single person that suffers with Alzheimer's disease, but also with the family that are taking care of the person affected by the disease. There is a whole other world affected by this, and being aware, raising the awareness to fight Alzheimer's disease can be, as we were saying before, faced from different point of view. By now, what I'm doing is putting my little piece of puzzle as a basic scientist research. I'm thinking in the future that I would like to keep doing my outreaching science job with expanding my skills besides the bench work. So let's see what is gonna end up. Obviously, I have several ideas. I'm applying for green card. So let's see when I'm going to get that, where I'm going to be in term. Let's fight Alzheimer's disease from a different point of view. But I want to stay in the neurodegenerative diseases because, as I was saying before, it's not just me studying a cell with a disease. Think about that. People are suffering, and it's not just the people that are suffering, it's the people that are around these sick people. And we need to find a cure. We need to find something to alleviate this situation for sure.
And that's fair, right? A person's professional career takes many twists and turns, and so I didn't mean to kind of put you on the spot there, but I do like that advocacy.
That's what you guys are doing, like trying to raise awareness of the disease and why we are studying this and why that is relevant for the disease, for the awareness. And that's very, thank you so much, Toffler.
It's very important. You may not want to answer this question, but was there people in your life that have Alzheimer's and dementia? Did they kind of influence you? Did you see the disease up close and personal? I did with my grandfather, but I was just wondering about you.
No, fortunately, I haven't seen anybody with Alzheimer's disease. There have been people with what that has been called senile dementia, but we are talking about times when we were saying before where the progress is going to bring us. Now we are talking about decades ago, and at that time there were not the tools for understanding that probably that was the dementia, Alzheimer's disease, but diagnosed, let's say no. Also, that was an interesting part, when there was not this simpleness in explaining me what was going on with that aunt.
Yeah, we didn't know. It was just, oh, that's old age.
Exactly.
Now you're from Italy. You're from Italy? Do you still have connections, probably with your family, but are there professional connections to people in Italy that you work with that are working in this area of Alzheimer's and dementia research? And just, how does that collaboration happen?
Yes, I am definitely in touch with them, with people that I've done my PhD with, my previous PI, principal investigator in Genoa. I am collaborating also with another group in Genova that does metabolic dysfunction. So yes, we have been in touch both personally. That's always nice, but also professionally. Also because me coming from Italy in the United States, I've applied so many other different and learned so many other different techniques that complementarity, like they know what I knew, but then I knew others. I learned other things, and then we collaborate. We can collaborate from other points of view with like diverse aspect of the research.
Yeah, because it's a global disease, and we need to, kind of work that and collaborate it. And I'm just kind of curious, in Italy, is there as much emphasis on this kind of research on Alzheimer's dementia, or do you see that the United States puts a little more emphasis in funding in that area?
Let's say in terms of awareness, I think the two countries can be comparable. Needless to say, but also the fact that people like me came to the United States to study the disease, but the Alzheimer, cancer, heart disease. The fact that here the fundings are bigger, are greater, so it's easier to perform, as we were saying before, cutting edge techniques, establish collaborations that can achieve that kind of assay you wanna make, and with that amount of money, which is still a good amount of money. And here in the United States, it's easier because of the fundings are different, obviously.
Yeah. So, you're studying kind of existing drugs that have been around for a while and its impact on the disease. What are kind of the potential implications of the findings of what you're doing with these existing kind of drugs on any kind of clinical applications in the future? Does this kind of understanding, will it lead to possibly new diagnostic tools? You talked about biomarkers. There would be diagnostics and possibly new therapeutics. Is that correct?
Yeah, correct. I mean, we know that besides the cognitive impairment in Alzheimer's disease, which is obviously the main thing we need to fight, there are characteristics like brain amyloidosis, we said several times, neuroinflammation. So we found that carbonic anhydrase inhibitors can actually revert this toxic event. And plus, we also know that carbonic anhydrase inhibitors help the clearance of toxic brain waste, such as amyloid beta, and thus reducing also the amount of amyloid around the vasculature, and therefore having a beneficial effect on the clearance. We may think also in the future to translate the drugs also in the combination with those already FDA approved drugs that currently are some of them partially working on both the accumulation of amyloid and on the vascular on the cognitive impairment. But we also know that the big, huge side effect of two of them, actually, then let's see the third of it, that it just have been approved a couple of weeks ago, if I'm not wrong, 10 days ago. One of the big side effects, I was saying, of the first two FDA approved AD drugs are these so-called ARIA, which are the amyloid-related abnormalities, vascular abnormalities, meaning they find once they treat the Alzheimer patients with this drug, they find a big accumulation of amyloid around the vasculature, trying to get rid of the amyloid. So the vessel become fragile and they break. Indeed, one of the, again, another side effect is these microhemorrhages. Now, our carbonic anhydrases are drugs that help and have beneficial effect on vascular fitness and help the clearance of amyloid. Thus, it can be thought one day to have our carbonic anhydrase inhibitors used in a parallel approach with the current FDA-approved drugs.
So do I understand that correctly? The existing drugs that have just been approved in the last year, couple years, that by cleaning out the amyloid out of the brain, that's actually putting stress on the vascular system and weakening it, making it fragile. Is that did I understand that?
That's it. In Deb. So besides, there is this dual approach of cleaning the brain, and several approaches actually. What I was talking here is because of the vasculature, and what I'm doing as well in the lab is studying what also the glial cells are doing. So degrading and like also physically phagocyting, meaning internalizing and degrading what is external and what is bad, amyloid plaques. For example, and so here there is this dual cell type that are involved in the cleaning of the brain. But yes, that's one of the big thing of the FDA-approved drugs. They clean the amyloid from the brain, stressing a lot the vasculature because the vasculature cannot support the such amount of amyloid that must be cleared that gets stuck around the vasculature. Therefore, vasculature become weak, sick, and leak, and creating this second side effect that are the ARIA cause microhemorrhages.
So this research that you're doing is very important and very timely because, as these other drugs get to market, get to the patient, having other things that may already be approved may actually, in conjunction with these other drugs, help so that it's not doing, damage while it's cleaning the brain. You're fixing one disease or you're attacking one disease and you're creating another problem somewhere else.
Right. Well, of course, that's the advantage of using FDA-approved drugs as acetazolamide and metazolamide are. But another big discovered that part of the study published in Alzheimer and Dementia is that not only carbonic anhydrase can revert the toxic phenotype of Alzheimer's disease, vascular, brain, blood vessel wise and inflammation wise, but also these toxic events are likely mediated by a specific mitochondrial isoform of carbonic anhydrase, which is called carbonic anhydrase 5B. So what's the next step? We are starting from an already FDA-approved drug with that specific pharmacological structure that inhibit several isoforms of carbonic anhydrases, but we know that in neuroinflammation and neurovascular dysfunction in Alzheimer's disease, the one that likely is the mediator, the mediator is the mitochondrial 5B. The next step is going to be pharmacologically create a new inhibitor that targets specifically that mitochondrial isoforms, because we know also that mitochondrial stress is a big thing in Alzheimer's disease, oxidative stress. Well, there is a big part played by mitochondria. So, yeah.
Is it going to be necessary to go into kind of clinical trials with the existing drugs that you're using, these carbonic inhibitors, these CAIs? Will they go into clinical trials along with some of the other things that are being developed for Alzheimer's patients, or will they not have to go through that clinical trial step?
We are trying actually to get them in clinical trial because we were saying before, they are FDA approved for AD non-related conditions. So that's very interesting because then the part of understanding what actually the carbonic anhydrase inhibitors already approved do in the actual brain of AD patients. But that's interesting, but that's nice also because we know that we know already all the side effects of this drug. We are using very lower amounts compared to amount of concentrations compared to the one that is known that can give some side effects. So we are on the good side. I told you, we are also working on parallel because we were saying before, the more the science goes on, the more it develops, narrowing down what could be a better target. So we started from carbonic anhydrases. Now we are going to a mitochondrial isoform of carbonic anhydrases. And then it would be nice to understand which are the cell type which are mainly involved in vascular dysfunction besides endothelial cells, glial cells, astrocytes, microglia, or smooth muscle cells, pericytes. This beautiful set of cells that orchestrate in tandem to have, in the end, having neurons to function. No?
Yeah. Are there researchers that are kind of in the same line of research, but are targeting dementias, maybe aphasia, Lewy body? Do you know of anybody that's kind of using the same research focus to look at some of the other kind of adjacent neurological diseases?
Well, someone in our lab is studying tauopathies, so dementia that are characterized by the accumulation of tau, which is still a hallmark of Alzheimer's disease, but it's associated with amyloid plaques. So he's studying a mouse model which normally it's characterized by a mutation which occur in frontotemporal dementia. So, but we are still the lab that we are studying the damage that tau could trigger to vasculature in the brain. And so we'd like to focus and understand is also tau, which is unknown for now, can trigger this kind of vascular dysfunction. And it's interesting because why we do vascular dysfunction? Because we know that at early stages of Alzheimer's disease, sometimes also when amyloid and tau load is mild, is already present. Is already present and is accompanied by inflammation. So that can be an early event to target to then facilitate the clearance of amyloid, ? having the vasculature that is more functional can ameliorate that part of the accumulation of amyloid or tau. So yeah, a lot of things to do, Deb.
A lot of things to do, and not enough time and not enough money. But that area of looking at the accumulation of tau is kind of an emerging area that needs some focus to understand that accumulation and its impact on the health of the cardiovascular system.
Yeah, especially nowadays that with Western diets, for example, so high fat diet, high carb diet, so also the part of diabetes. And we know that there is a higher average of Alzheimer's disease in the Western countries. So nowadays is fundamental, essential to understand if there are mediators that are converging in the two pathologies that are like in bidirectional, in a bidirectional way, exacerbating one or the two pathology. And that's what we're doing as well in the lab. So studying heart disease in a model of Alzheimer's disease. No? So the goal is to understand whether the presence of transgene normally characterized the Alzheimer's disease can also affect the heart function or pathology. So yeah, that's what we are doing. Plus, we are also studying the outcomes on vasculature of animals that are transgenic for Alzheimer's disease in combination with cardiovascular risk factor such as homocysteine and L-NAME, which is known, it's a compound that increases blood pressure. And we want to know if these cardiovascular risk factors can make the brain more prone to aggregate Alzheimer's disease and therefore to accelerate the pathology. So yeah, a lot of interesting things are going on in our lab.
Yeah, it just makes sense, right? What's good for the heart is good for the brain. That's kind of the basic. Now we need to understand why and the kind of the mechanisms there. Looking into the future, what are you most hopeful for? If you kind of step in and say 5-10 years from now, what do you really hope we're able to either know or be able to do?
I'm going to give an answer, a global answer. Let's say that as a young scientist and a woman, my big hope is to see a world in the future that is going to more worried about the global changes, starting from health, obviously including Alzheimer's disease, but also to global warming. And hopefully where social and death and gender disparities are not going to be there anymore. How? With a combination of knowledge and technology. That's what I wish the most.
Yeah, that's great. What do you think will be the big technology drivers in the next? You mentioned a couple, but is that imaging, AI, computational capability, or do you think that's going to be kind of a big driver to accelerate some of the learning?
I think they are gonna be a big accelerator about helping. That's the big thing, helping, assisting the knowledge of the human mind. Obviously, that's a big thing that we're never probably gonna get big advancement, scientific advancement, if there is not a human mind behind that, algorithms that generate, that develop the question that the human mind have asked. So let's say that I really believe that these artificial intelligence and machine learning are great partner of us, human scientists, let's say that. But for sure, their help, their assistance are gonna give us a kick. If you just think the kick, scientific kick that we had in the last 20 years, no? It seems like 100 years ago in terms of the how fast it's going. So I'm not doubting that is gonna be a big help, but as everything must be like limited and help to be developed. That's what I'm saying.
Yeah, the human is still very, very important in that collaboration, if you would.
Definitely yes. No, definitely yes. I was at this conference a couple of months ago. Like I'm part of this association, Italian association, and we organize cultural events, and we host this guy that does artificial intelligence, and he was saying that there is always gonna be a big percentage that is needed from the human being, because there is the innovation that can be given by the artificial intelligence, but the trick, the different point of view that the human is gonna give is never gonna be comparable to the AI.
Yeah, I agree with that. I think it will be a wonderful tool and a wonderful kind of partner, but we still need that that kind of that randomness that the human brain brings, that challenges in the different perspectives. So I agree with that. So this has been wonderful, and you've really expanded kind of that view of, wow, we're really broadening our perspective of what's happening in the brain and how this disease. So I appreciate that. You talked a lot about collaboration. How would people, if they want to collaborate, find more, how would they get in contact with you?
Obviously, they can reach me out on LinkedIn. My name, Elisa, and my last name, Canepa, or you can reach out to me to my work email, my name dot my last name, elisa.canepa@temple.edu. And yes, that's it. Thank you so much, Deb. It was so much fun. And hopefully, I'm happy people are going to reach me out, and I'm going to be glad to explain further about my research, or why not expanding my scientific network, or who knows? Who knows? It's always a pleasure to have my research shared with whoever. So that was just a pleasure for me. Thank you so much, Deb.
No, thank you. And thank you for your passion, and thank you for that focus of awareness, and that we just need to make people aware, and all the things that we need to do. So thank you very much.
Thank you.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.